So we will have a question and answer session at the end when all of the speakers will come up. So if you have any questions, write them down or remember them, and you'll have an opportunity to ask. Thank you, Jonathan, for this a very interesting talk on a very controversial, as we know, controversial topic. So our next speaker is Professor Nir Ohad. He's the head of the Manna Center in Tel Aviv University, and he will talk to us today. His title is From Flower to Flower, Leveraging Basic Science Towards Sustainable Agriculture.
It's a pleasure being here and to give this talk today. So I'd like to start with a news piece from about 2,000 years ago. What we're seeing here is the move of Abraham down to Egypt. And the piece of news says from the Bible that there was a famine in the land and there was no choice for Abraham but to go south to break the famine. So, as we said, there's no new under the sun, there's no new things. We're still coping with the same problems today. And just recently, Israel encountered the problem of other countries. So if one country is safe and secure, it doesn't mean that it is not attached to the neighboring countries. See what happens in Sudan or in Eritrea in 2007 when tens of thousands of people were fleeing because of instability. If you just calculate the amount of people that arrived to Israel into percentages, which was about 65,000 people, and you'll translate it into Chinese population, they will encounter about 15 million refugees coming into their doors. But this was not the end. We had in 2009 the Arab Spring due to instability and food problems, which then continued into local wars, which we're still encountering around us. Just look around what happens in Libya, Syria, Iraq, Lebanon. And the outcome is, we're already all familiar with it. This is the outcome. You know, people are fleeing to find safe haven, and secure countries are no more secure because look at the map of 2015. More than 1 million immigrants are knocking on the door of Europe. Mediterranean, no Mediterranean, they'll cross, right? So we have to think globally. It's not only our own problem, it's a global problem. So Israel encountered also some sort of riots. We had the food issues, the Mechat HaCottage, which we all remember when price of food were rising. And it's a local problem as well. So we need to treat first our own problems, but to think globally. Yeah, we went out to the streets because of the rise of price of food. So we need to think, what can we do to ensure our food security in the future? So the challenges we have globally, not only locally, are of course the increase of global population, and FAO has addressed this issue and updated it. We're considering that we'll have in 30 years 11 billion people, not only 9 billion people as it was expected before. We have decrease of arable lands, which Jonathan Gressel has touched upon. And if we look at the increase of crop yields, this is something which can be dealt, but it's very slow increments because science has its own pace. So if we look globally today, perhaps half of the world population is not secured. And some of them, though they have the source of food, are even overfed. And this is a matter of choice. So our mission globally is to have a significant contribution to Israel and worldwide security, and this is what the Manna Center for Food Safety and Security is trying to do. So today I would like to describe to you a few of the research topics that are being studied here in Tel Aviv. We touched upon the general understanding of what food security is, to provide all people at all time affordable and nutritious food to support sustainable life and healthy life, and this is one needs to do. So the talk today title was From Flower to Flower. Where's our basic food coming from? It starts from something that I'm studying already for the last 25 years, and that is plant reproduction. Now, plant reproduction starts when a pollen grain, you see these yellow dots fall on the stigma, and then penetrates in to fertilize an ovule. So we'll have here within this ovule a single cell, which is then fertilized, giving rise to embryo. Yes, plants have embryos. This is what most of us eat. This is the source of our nutrition. In addition, plants have a second type of an embryo. It's a hidden embryo. It's called endosperm. This does not grow to the mature plant, but this is a source of nutrition for the seed and seedling. So endosperm provides carbohydrates, and embryos provide proteins and fat. And those were meant initially by evolution to support the growing seedling, but we have learned how to use them, and this is the source of our nutrition. So our world nutrition is based on plant biology. This is what we need to understand in order to improve our food security. Now, every nation and every region has its local food variety. We're emphasizing Mediterranean diet. And indeed, in this region, the main source for carbohydrates is wheat. Aaronsohn has identified the source of wild wheat from which it was domesticated probably 10,000 years ago, maybe even more, the mother of wheat. And when he identified it, he already then wrote at 1910 that the cultivation of wheat may be revolutionized by utilization of these wild forms. And I'll give you today an example how modern research can actually do this. So on the left-hand side, you can see the wild wheat, and on the right-hand side, the domesticated wheat. Even if you're not a plant biologist, you can see the major differences between the two. And when you treat or you breed, you take from the wild the one species to generate the other species, what you usually do, you do a cross. And this is how our forefathers did breeding. They simply crossed between one plant and another to look for the better plant. When you actually do that, you can simplify it into looking into a pool of genes. So each plant has, in wheat, about 1,700 books, and each book about 1,700 pages. And now, like a pile of cards, you just mix and shovel between those sets of genes. Eventually, you get a hybrid between those two sets of genes, and you start to look into this pool of books and pages, which if you would pile them will reach a 20-story building, if you could actually physically do that. And what you're actually looking at the end is this particular one gene of interest, maybe the resisted gene, or to take out a component which can be toxic. So this is how breeding was done, just by cross and selection, which takes a lot of time. This is why those slopes of increased food yield take so much time. So here's again the wild variant of emerald wheat, the one that Aaronsohn really discovered, the one that was used to domesticate what we eat today, the durum wheat or the common wheat. And you can see the differences, but this took thousands of years to be reached to the point we see today. So how can we make a faster advance? And how can we accommodate new problems, such as emerging diseases? This nice rust color, this is not something that you'd like to see on your field. This is a pathogen, this is a fungus that when it attacks the wheat will kill it. And it's actually a disease that is spreading from Central Africa towards our region. It hasn't reached yet, but if it will come, there is no cure to it until recently. So what can be done? So here's one example of how you can use modern technology. Members of the ICCI, the Institute for Cereal Crop Improvement here in Tel Aviv University, which are also members of the Manna Center for Food Safety and Security, Dr. Asaf Distelfeld and Professor Amir Sharon, the head of the ICCI, together with Dr. Gil Ronen from Energene, has done two things. First, regardless of this project that was done with Energene, they have been able to map a particular gene that is present in the wild species and then to cross it back into the cultivated species to confer resistance. So this is the first step. This is a major breakthrough. This project was financed by the Bill Gates, Melinda Gates Foundation, meaning that whatever they'll generate is going to be contributed to the world freely. Now, this is not a simple thing. We are looking into solving problems, and we need to do research that will help society not only for profit, and this is exactly what you do. Now, if you consider that Israel is not a major producer of wheat. We actually import most of our wheat. But wheat was never grown in north of Canada or Central America. It was imported from here. They had to adjust wheat growth to their conditions. And we have the genetic resources here from the wild species that are thousands of wild species that we have in our collection to exactly that, to confer resistance to different disease. But even more, now this consortium with the ICCI and members of the wheat community and this new startup company were able to sequence the entire genome of the wild wheat and recently the domesticated wheat. Now we have the new tools to move forward much faster, to start and do improvement. This is from Asaf Distelfeld research from the ICCI, show us how you can advance from right to left in increasing grain size. This is important because we won't have more land to grow, but we could increase yield if we do the right steps. So far we're talking about making the seed. And maybe we have now the recombinant tools, not necessarily through GMOs, but genetic tools and molecular tools to do a faster breeding using the knowledge we have. But now I want to transit to a new topic, something that I'm involved in. This is my own research. For years, we've been looking to improve seed quality by looking for particular genes that can help us to understand the make of an embryo and then a plant. This was a hard task, and recently we have moved into a model plant. What is a model plant? In this case, it's a moss plant. Moss have evolved more than 400 million years ago and served an extra model, superb model, to understand plant reproduction. How does a moss reproduce? Well, it starts with a small tissue, which is a filamentous tissue, then grow a stem and leaf, and eventually it fertilizes itself through the merge between male, these are the male gametes, and the female gametes. Here you can see the egg cell, a single egg cell of a moss. Now, upon fertilization, an embryo will grow and eventually a mature plant, which makes the next generation, the spores. And here you can see many, many spores. So what we've identified through genetic screening is a particular gene. So we've isolated a particular gene that when we express it in the plant without reproduction, it generates the mature plant through an embryo. So we've bypassed the need for fertilization. Well, you say, what's the relevance of a moss, which evolved 400 million years ago and still with us, to agriculture? Well, here's the key issue. What we've basically found was a single very important factor, a genetic factor, that can overcome sexual reproduction. Why is that so important? Because, as I've just told you, when you're going from a flower to a seed to a fruit, you need to involve cross between gametes, male gametes and female gametes. And usually, when this process takes place, we get a mix of seedlings, of seeds. And they have different traits. They won't grow entirely to the same height. They won't mature to the same time. So you need to adjust all your cultivation towards that variation. But what if you can have all seeds alike? This is what we called apomixis. This is what we can do with such a single gene. If we can transfer this technology from the moss into the flowering plants and generate ideal seeds which are all alike, just as the same of the mother plant. This is the future of agriculture. This is what the major companies are looking for. And then we will move forward. We'll have the genetic material. We can have the best content of the seed. But when we reach the field, and here we have an example of another project, Yotam Laor from the geography department under the lead of Professor Eyal Ben-Dor. So he's just collected potatoes. One of the major issues about food security is what Elliot described to us, is about 30% loss in non-developing countries from the field to the market, and in developing countries from the supermarket down to the consumer. That's where we lose most of our products. And this is an issue, of course, also for potatoes. So what Eyal Ben-Dor and his team are doing is to devise spectral analysis to identify rot of potatoes before they even start to show the symptoms, just by looking at the light emitted from those particular potatoes. And then once they can calibrate this in the lab, they can use satellites, airbornes, cameras, or any other device to identify that already in the field before those problems occur. So this is just one example how we can move forward from doing the grain product to the growth and then to the food loss. So we've started with the grain, with the project I've described of the ICCI. We moved to food loss, and then this is what we're actually doing in the Manna Center, is trying to bridge the different disciplines over the university here and to look at the entire food chain, or if you wish, from field to fork, or מהשדה אל השולחן. And you'll hear later today Dr. Ram Fishman of how we can implement new technologies once we have our ideal seed, once we have our ideal conditions to grow the plant, how we can implement it, how we can support mainly small farm holders in developing countries to implement those new technologies. But we need to think even further downstream and to look into new opportunities for generating food. And what else if not looking into our aquatic environment? So fish and other products as algae and marine life can serve as a protein source. And look just what happens in the States for the last 10 years, the increase or the import of fish, mainly tilapia. Tilapia is the second most consumed fish around the world. Who are the major growers? China has increased interest in products and export of tilapia, as well as Egypt and other countries. Israel, relatively to what is produced in the world, well, tilapia, as I said, is the second consumed fish in the world. In the States, it's about 4.5 million metric tons used per year with a value of about $7.5 billion and expected in 2030 to reach 7.3, almost double million metric tons in the world. And the US consume about 230 million tons annually of tilapia only. So tilapia is a major protein source, but not only, it serves as algae and mosquito control, and it makes the habitat for shrimp growing in fresh water. Now, Israel, as I said, is a small producer, only 1,000 tons per year, but, so we usually grow our tilapia either in freshwater ponds or in the Sea of Galilee, but in recent years we had a decrease in product, and it turns out the fish were sick. And a team from Tel Aviv University affiliated to the Manna Center, led by Professor Eran Bachar, Marcelo Ehrlich, together with the Vulcani Center of Veterinary, the Qimron Veterinary Institute, work done by the first Cameroonian student, PhD student here in Israel. They have actually isolated the cause for this disease, which was a virus. So in 2014, they identified that the cause for this disease is a virus, but this year they've actually isolated the virus and published it in a very high-profile journal describing this virus. Now, why is this so important? As we can see, tilapia trade is across the world, and the virus can be born even when the fish already is dead and can contaminate other resources. Actually, we know that the virus is present already in South America, not only here. So if we are looking for the increase of tilapia produce, we're somewhere down here, 2010, by 2050, it should double the amount of produce where tilapia is going to be a leading product. We need to devise new strategies to fight such diseases, and we have the tools. So fish vaccine is the way to do. Now when we have the virus isolated and the genome isolated, there's a mode or method to try and develop those vaccines, which is underway. So I've shown you today that by enhancing research from different disciplines, geography, life sciences, and social sciences, as well as public policy, bring together with research, education, and outreach, as we're doing today in this meeting, what we're hoping to do is do exactly this, to make a better world and produce our knowledge to enhance food security. And I would like to leave you with this, as Elliot said, with a seed of thought and a seed of information to take with you. It's a 1 carat, very expensive, very precious piece of information, which comes from the seed of the carob tree. And those of you who are interested, I have some examples. So I thank you all for your attention and look for an exciting meeting. These are the sponsors and collaborators of our own research. Thank you very much.